I hope you're having a good weekend, and welcome to the PowerPoint on meiosis. This is the second type of cell division that we'll be studying. And as we mentioned with mitosis, this is really about nuclear division, not so much the cell division. That happens right after the events of meiosis and mitosis occur. So in meiosis, a single diploid cell gives rise to 4 haploid cells. Just a reminder, a diploid cell is the one that has the normal chromosome number for the species. In humans, that number is 46. Haploid cells have half the number, so our sex cells, the sperm and egg cells, have 23 chromosomes. This occurs in 2 different stages, meiosis 1, which is a little bit quirky, and meiosis 2, which is very much like mitosis. PMAT still applies, but now it's doubled. So you go through prophase, metaphase, anaphase, and telophase twice. The first time we call it meiosis 1, prophase 1, metaphase 1, anaphase 1, telophase 1, and then we have meiosis 2, so it's prophase 2, metaphase 2, anaphase 2, and telophase 2. In prophase 1, just like with mitosis, DNA replication occurs. This occurs during the S portion of interphase. During prophase 1, the homologous chromosomes pair and form a tetrad. This is different from what we saw in mitosis, so they're paired up with their opposite number. This step is unique to meiosis. They call this synapse, the time when they are paired up, and while they're doing this, crossing over can occur. Crossing over is when bits and pieces of the genes from one chromosome are swapped with the other one. And this increases the genetic variation, which is a very good thing for the diversity of a species. And if I'm going too fast, of course, you could pause at any time. Prophase 1 continues, the nuclear membrane disappears just like it did in mitosis. A kinetochore forms per chromosome rather than per chromatid. A kinetochore is what we call the sticky portion of the centromere that attaches to the spindle fiber. And there you have it. I didn't mention that in mitosis, and I'm not even sure why I put it here. I don't think you'll see that on the regions. In metaphase 1, the tetrads, now composed of 2 chromosomes or 4 chromatids, align at the metaphase plate, just like before. They're lining up right down the middle. The centrioles and the spindle fibers pull them to the middle, but they're not lined up one way or the other, meaning all of dads are on one side, all of moms are on the other. They're all mixed up. So this is another way happening that is increasing the genetic diversity, making sure that the haploid cells that are formed at the end are all different from each other. In anaphase 1, the chromosomes split apart to the separate poles, but the centromeres between the sister chromatids stays intact, unlike anaphase 1. Telophase 1 happens just that fast. In telophase 1, the nuclear envelope might reform depending on the species, or the cell might just go directly into meiosis 2. So all those other things we learned about in telophase, the nucleolus reappearing, the nuclear membrane reforming, that may or may not happen. We are now into cytokinesis 1. This is when the cells physically separate and you wind up with 2 daughter cells. We are now ready to go into interphase 2. And a big difference between interphase 2 and interphase 1 and interphase in mitosis is there is no replication of DNA during this phase. Let's take a look at the animation. Yeah, what they're showing here is the chromosomes replicating to make another copy of itself. You know what? This video is not that good. I'm going to skip it. We're going to go back to the PowerPoint. Square dancing. There we go. So meiosis 2 proceeds exactly the same way as mitosis. The steps involved, however, have a 2 behind them. So prophase 2, metaphase 2, anaphase 2, and telophase 2. Just like mitosis, meiosis 2 is followed by cytokinesis. The end result of meiosis 2 is 4 genetically different haploid cells. And this is why, if you have siblings, you are not carbon copies of each other. Each of you has a very distinct genetic fingerprint that is only yours. Gametogenesis is the production of gametes, and it's different for males and females. In males, the process is called spermatogenesis. In females, the process is called oogenesis. This is what it looks like in males. The spermatogonium cell is produced by mitosis. It's like any other somatic cell in your body, but then it goes on to produce sperm cells through meiosis. And in the end, you wind up with 4 functional sperm cells, each with 23 chromosomes and are genetically different from each other. In oogenesis, again, the primary cell that starts this is formed through mitosis, but then after that, meiosis occurs, and you wind up with 1 egg and eventually 3 polar bodies. The polar bodies in humans are not used for anything, they're reabsorbed by the body. And there you go. See you on Monday.